In the last lecture of our biology series, we're going to go into genetics and evolution. We'll start off with some fundamentals. There are ways to quantify expression, and let's go into some details of expression. An allele is an alternative form of a gene. There is dominant and recessive. A dominant gene requires only one copy for expression, whereas a recessive requires two. Genotypes are the combination of these alleles at a given locus. Homozygous, meaning that there's two of the same, heterozygous, meaning there's two different, and hemizygous, where there's only one. Phenotypes are the observed manifestations of a genotype. There are varying patterns of dominance, three in particular. The complete dominance states that one dominant allele and one recessive allele are present. Because of this, the expression pattern is going to be noted. This is the one expression pattern that we generally think of. When there's one dominant allele, it will take over and be the expressed trait. Codominance is when there's more than one dominant allele. And incomplete dominance is when there is no dominant allele. Heterozygotes that are expressing incomplete dominance are going to have intermediate phenotypes, such as the flowers below. Flower colors in this particular species are not noted as being dominant in any way, shape, or form. Therefore, heterozygotes are going to have a pink shade because the white and the red genes are not completely dominant. Now that we've talked about expression on the individual scale, let's talk about them on the population level. Penetrance is the proportion of a population with a given genotype expressing that given phenotype, whereas expressivity is how varying phenotypes manifest of a given genotype. Examples are listed below. Variable penetrance, variable expressivity, variable penetrance, and expressivity are noted. Mendel is commonly called the father of modern genetics, and there's two laws that he talked about when he's talking about gene expression. The first is the law of segregation, which states that any organism has two alleles for each gene. Meiosis will yield one allele for any given trait in a gamete in sexual organisms. The second law states that independent assortment is present. The inheritance of one allele does not influence the probability of inheriting a given allele for a different trait. So, if I were to get the gene for brown eyes, that would not affect my ability to get genes for black hair or blonde hair. Each gene is inherited independent of each other. Now that we've talked about some fundamentals, let's go into changes in the gene pool. One of the most common ways that the gene pool changes is via mutation. There are different kinds. Point mutations are simply when one nucleotide is substituted for another. Frameshift is when three-letter reading frames are moved. A silent mutation is one in which there is no effect on the transcribed protein. However, an accumulation of silent mutations can lead to changes in the future. Missense is when the substitution of one amino acid is switched for another. Nonsense is when a stop codon is substituted. Insertion and deletion is when you add reading frames or transcription points. This will shift the reading frame. And deletion is when large segments are lost. A duplication is when a segment of DNA is copied multiple times by accident. An inversion is when this DNA segment is reversed. An insertion is when a segment of DNA is moved from one chromosome to another. And lastly, translocation is when a segment of DNA is swapped with a segment from another chromosome. Mutations will often lead to different types of expression patterns and phenotypes, leading to unique formations of organisms. There are also population effects when we talk about changes in the gene pool, and the three in particular are listed below. Genetic leakage is when the flow of genes between species through hybrid offsprings occur. If you've ever seen a liger or a lion-tiger hybrid, this is an example of that. However, you can't say that this is a fully functional organism because that heterozygous offspring is not viable. Genetic drift is when the composition of gene pools change as a result of chance. And the founder's effect is when there is a bottleneck that isolates a small population. The inbreeding will increase certain prevalence of homozygous genotypes. This is going to drastically alter the gene expression in the isolated populations, and at worst will lead to speciation. Now that we've gone over fundamentals and the changes in the genotype, let's talk about how we can analyze genetics on a scientific level. We'll start first with Punnett squares, which I'm sure you're all familiar with. This is to map out the alleles from the parents and to look at the offspring that they're going to produce. An example is listed to the bottom right in the form of Mendel's pea plants. I'm sure you've noted more complex Punnett squares with multiple alleles being taken into consideration. In general, the MCAT's not going to go that far with things. Recombination frequency is listed as the likelihood of two alleles being separated during crossing over, the equation for which is listed below. Hardy-Weinberg principles state that populations meeting certain criteria will have an allele frequency that remains constant, which is noted as the Hardy-Weinberg equilibrium. Take note that the Hardy-Weinberg equilibrium makes some assumptions. For starters, there's no selection of individuals, no mutations, no migration, a large enough population, and completely random mating. As you might suspect, these are some very serious assumptions and not ones that are commonly seen in the natural world. It's impossible to talk about genetics and speciation without talking about evolution, which is a theory that the late Charles Darwin pioneered. There are some fundamental principles when you talk about evolution, starting first with natural selection. This states that variations will increase fitness in an environment, and this will allow for more success. Success in terms of evolution is the ability to reproduce. Neo-Darwinism is basically a newer form of Darwinism that accounts for mutation and recombination as vessels for success, along with natural selection. Inclusive fitness considers an organism's success by these following criteria: the number of offspring produced, success in supporting offspring, and the ability of the offspring to support others. Lastly, the survival rate of these offspring and these given relatives. If you can survive and reproduce, your genetic material will pass on, thus leading to success. There are equilibrium patterns which consider evolution by a very slow process with intermittent rapid bursts, and there are different types of selections. Stabilizing selections will keep phenotypes in a narrow range. This is largely based on environmental factors that are perpetuating these sorts of phenotypes. Directional is going to push phenotypes to an extreme. So if an extreme environment change occurs, then only individuals with certain extreme traits are going to be able to reproduce successfully. Disruptive is when that these expressions split and move toward different phenotypes, which may lead to speciation. An example of this would be if you were to look at birds that were eating seeds or bugs. If this split were to perpetuate further and further, then birds with beaks more suited for eating bugs would eventually speciate from birds that were more suited to eat seeds. An adaptive radiation is the rapid emergence of multiple species from a common ancestor, each occupying an ecological niche. This occurs in huge bursts, and you're going to note this very commonly in our biological history. What is a species? I've mentioned species earlier in the lecture, but what does it constitute? It's the largest group of organisms capable of breeding to form fertile offspring. They're also reproductively isolated by pre- or post-zygotic mechanisms. An example of a pre-zygotic mechanism would be being unable to fertilize and successfully create a zygote. Post-zygotic mechanisms could be involved with after the formation of a zygote. It's possible that the mother is unable to host this zygote, and thus the pregnancy fails. The liger example I mentioned earlier is an example of this. You can reproduce a tiger and a lion, but ligers cannot reproduce. Speciation has led to the formation of a lot of very different types of animals, an example of which are dogs. Dogs are descended from wolves and coyotes, and they're reproductively isolated because of the speciation that has occurred between the two. There are varying types of evolution patterns, and there's four in particular we'll go into. The divergent evolution states that when two species have a common ancestor, they evolved differently. So, from a common ancestor, they went their separate ways. Parallel evolution is when two species sharing a common ancestor evolve in similar ways due to similar environmental pressures. So it's going to be in parallel to each other. Convergent evolution is when two species not sharing a recent ancestor evolve to become more similar due to similar pressures. An example of this would be wings. Butterflies are obviously not terribly related to birds. However, they both evolved some sort of wing-like structure because of similar pressures. The molecular clock model states that the degree of difference in a genome between two species indicates the relative amount of time since the two species broke off from a common ancestor. As you would guess, because of this model, it can be stated that eventually, at some point, we were all from a common ancestor. Here's a visual example of these different types of evolution patterns. Okay, so that'll finish out the biology section of our MCAT prep series. Take these to good use. If you remember, there's an entire section of the MCAT that is dedicated exclusively to biology, so studying these subjects and having them to memory is going to be very important for your success on test day.